Welcome. Very basic and important question. What is analytical error in the pharmaceutical analysis? An analytical error is any deviation between the measured result and the true value due to flaw in method, equipment, or analyst performance. It affects accuracy and precision, leading to OS that is out of specification results. Reference ICHQ to R1 and USP chapter number 1225. What is percentage error? As per ICH Q to R1, percentage error is equal to measured value minus true value divided by true value, and after getting the answer, we multiply it by 100. What are common sources of analytical errors? The common sources of analytical errors are instrument errors, sample preparation errors, method errors, environmental errors, and human errors. What are error by analyst? As per WHO GMP and EU GMP Annex 11, incorrect pipetting or weighing, wrong dilution factor, transcription mistakes, and not following SOPs are the error by analyst. What are error due to equipment? Uncalibrated balances or HPLC, worn out HPLC columns, UV lamp instability in spectrophotometers, and software integration errors are the common errors due to equipment. Reference USP chapter number 1058 and FDS 21 CFR part 211. What are errors during sample preparation? Improper homogenization, contamination, incorrect solvent selection, and degradation due to light or heat exposure are the errors during sample preparation. Reference ICH Q1A and USP chapter number 659. What is error in method selection? Using a non-validated, non-specific, or non-robust. Poor resolution in chromatography, matrix interference in spectroscopy, and incomplete extraction in dissolution. Reference ICHQ to R1 and USP chapter number 1225. What is difference between systemic and random errors? Systemic or bias errors are consistent, reproducible deviations, example calibration errors, whereas random or precision errors are unpredictable fluctuations, for example, electronic noise. What is accuracy? The closeness of a measured value to the true value is accuracy, expressed as percent recovery in validation. Reference ICHQ to R1. What is precision? Precision is the degree of agreement among repeated measurements under same conditions. Repeatability or intermediate precision. Reference ICHQ to R1. Very commonly asked question, what is true value? An accepted reference value is called true value. For example, from a pharmacopoeia standards or certified reference material. Reference USP chapter number 11. What are random errors? Random errors are unpredictable fluctuations due to noise, environmental changes, or operator variability. These are often minimized by replicates and controls. Reference USP chapter number 1010. What are methods for minimizing errors? The commonly implemented methods for minimizing errors are calibration and maintenance of equipments, method validation and verification, training and SOP adherence, and control charts and statistical checks. Which guidelines are referred for analytical errors in the pharmaceutical analysis? ICHQ to R1, USP chapter number 1225, FDA's OS guidance, UGMP Annex 11, and 21 CFR Part 211 are the guidelines which are very commonly referred for analytical errors in the pharmaceutical analysis. What is the role of control samples in error reduction? Control samples like placebos, reference standards help to detect method or instrument bias. They must be analyzed alongside test samples as per USP chapter number 11 and ICH Q2 R1. What is gross error in pharmaceutical analysis? Gross errors are major mistakes, for example, sample mix ups, incorrect units, instrument malfunction, leading to invalid data. They require immediate investigation and invalidation as per FDA's OS guidance. How do you document analytical errors? Analytical errors must be recorded in deviation logs, OS investigation report as per FDA's OS guidance, and CAPA, that is corrective and preventive action systems. Don't forget to like, comment, and share. Subscribe to channel for more informative videos related to our pharmaceutical industry.